Yeah. Yes, you can start, Krishnanjali.
Okay, sir. Okay. Good evening to. Sir, am I audible? No.
Yes,
Hello.
yes, you are audible.
Okay, sir. Good evening to one and all. Dr. M. Deshmukh, sir. And today's our speaker, Dr. Vaishali Dange, ma'am, and all other participants. I welcome you on behalf of Microbiology Society. So today's speaker for our PhD webinar series is Dr. Vaishali Dange, ma'am. Currently, she is working as associate professor at Institute of Bioscience and Technology at MGM University, Aurangabad. She had completed her PhD from Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. Also, ma'am has 18 years of experience, and also she has published various research articles in reputed journals. Also, she has designed syllabus for MSc Biotechnology, MSc Microbiology, and BSc Biotechnology courses at IBT MGM University, Aurangabad. Also, she has organized many workshops and webinars. She had attended and participated in various national and international workshop, conferences, seminars. So now I request ma'am to start her presentation.
Yes. Thank you so much, Krishnanjali.
Thank you, ma'am.
Yes. Yeah. Am I audible, everyone?
Yes, yes.
Give me one. Yes. My research topic is on the on production optimization and clinical kinetics and clinical importance of microbial L-asparaginases. Actually, L-asparagines is the enzyme which is used in cancer treatment. So this enzyme, which is having a tremendous applications in the medical biotechnology as compared to the general biotechnology. So in my research, I have done the production with the help of different microorganisms, its optimization, and the kinetics of the enzyme. With that, I have done the applications of this enzyme in vitro uh, in vitro treatment. That means in cancer cell line, with the cancer cell line. So here we go.
Yes, you can change the slide.
Yes. So, so let us talk about the introduction. Actually, it is used for the treatment of the cancer patient. So it fight against the patient which is suffering the leukemia. So acute lymphoblastic leukemia is the cancer disease, and for that purpose, this enzyme that is useful. So the patient which is suffering this type of the cancer, we can treat with the help of the L-asparagines. So what the L-asparagines that will do? The L-asparagines, actually the reaction behind that is the L-asparagines hydrolyze the L-asparagine into aspartic acid and ammonia. So the aspartic acid, which is beneficial, or the asparagine, which is beneficial, which is present inside our plasma pool, and that is also used for the acute lymphoblastic leukemia. So, but the thing is what? If the L-asparagines can be given, the L-asparagine cannot be synthesized in the cancerous patient. Okay, so it means what? Ultimate the death it will be occurred. So the introduction part is what? The enzyme which is can produce with the help of the microbial sources that we can treat as a anti-cancer drug. Now the reaction, so biochemistry point of view, you can see that this is the L-asparagine enzyme which can hydrolyze the L-asparagine into aspartic acid and the ammonia. So this is the treatment that is the biochemistry part of this enzyme. So here we can see that there are the different isolates that I have screened out for the production of these L. aspergillus. So I have screened out different bacteria, different fungus, different actinomycetes. So from the population of that different microorganisms, I have got the 26 bacterial isolates, 17 fungal isolates, and 10 actinomycetes, which will give the higher production of actinomycetes. You can see in this pie chart. Now, after that, I have done the screening of my isolates with the help of the different plates. So you can see here, these are the plates which is showing the positive L. aspergillus production, right? So these plate one, it is showing that whether the enzyme is it is produced by the culture or not. So you can see in the plate and you can see in the test tube. So in the test tube, I have done the secondary screening of this enzyme. Next, now this is my the activity that I have done during my research. So these are the bacterial activity, the isolate which is showing the enzyme activity. So 26 bacterial isolate I have screened out, and from that I found only one that I will choose only one L. aspergillus best producer. So here you can see that this will give the high value, that is high enzyme activity, as compared to the other type of bacterial isolates. Next, these are the fungal results that you can see. So here SV4 that I have given the code name because preliminary I have not done the identification. So first of all, I have isolated the different type of the isolates, and after that I have done the identification. So SP4 is the code name for my fungal culture, and you can see that it is giving the high enzyme activity as compared to the other isolates. And this is the result that I have got for the actinomyces. So here you can see that the enzyme activity is the highest as compared to the other actinomyces. Actually, I have got the 10 actinomyces, but that the Act 1 is showing the high activity. Now this is the time to give the name. So after that, after screening out of the different type of the isolate, I have go for the identification. So bacterial SPB1, which is showing the highest activity, in that isolate, I have screened out the technique which is called as the 16S rRNA DNA sequencing, which is that is performed in the NCCS Pune. Next, actinomyces, that is I have done with the technique that is slide culture technique, and the name is for that actinomyces is the Streptomyces species, and for the bacterial isolate, the name is the Alcalogens faecalis. This is the new bacteria that I have found, and it is having the low research at that time because I have done the PhD in the duration 2009 to 2012. Next, the fungus that I have identified with the help of 28S rDNA sequencing. Actually, I have given the sample to the Cedris lab, which is situated in the Ahmedabad. So these are the 3 isolates that I have choose, and I have done the identification as per the technique. You can see here. Next, yes. So these are the results that I have got with the Cedris lab. So here you can see that lane 1. That is gel electrophoresis. So in that you can see the gel one, it is showing only one lane, and the lane 1 it is showing the only one band, whereas lane 2 it is showing the different band, which is having the different best pair. So I have compared with that, and I have given the name as a Aspergillus niger. So for that I have done the phylogenetic analysis. So here you can see that this is the phylogenetic analysis. And next, next, I have done the cultural conditions. So what are the conditions which is applied for the high production of the my enzyme? So here, if you see that the enzyme that first I have go for the activity, whether it is produced by the isolate extracellular or intracellular. So I have found that in violet color, if you can see here, the L-aspergillus activity is more as compared to the intracellular. So, but in that I got the Aspergillus niger producers, which will produce the high enzyme activity. So I have selected that Aspergillus niger um, uh, for the um, further studies. Next, optimization of the cultural condition, because I don't know whether the enzyme it will grow at what pH, at what temperature, at what incubation period. I don't know. So I have done with the help of the different incubation time. I have applied for the different type of the culture. So these I have choose for the three cultures. So incubation period, if you can see that the graph, the enzyme activity will increase, but it will begin with the 24 hours and it will finish till the 96 hours. So 4 days. In 4 days, I have got the maximum activity as if you see in this graph. Next is I have done the pH. So optimum pH for the production of my enzyme is 9. So 9 is the basic pH, not like a alkaline condition, but it's nearer to the basic at where I have got the more production of the enzyme. Next, the temperature, so 30 degrees Celsius, yeah, 37, up to 37, 30 to 37 degrees Celsius is the optimum temperature for the production of my enzyme. So here you can see that again, Aspergillus niger showing the high optimum enzyme activity as compared to the other two isolates, that is Streptomyces species and the Alkalogens faecalis. Now, next I have did the conditions what is applied, whether the fermentation condition, stationary and the shaking. So in that I have found that the stationary condition is more favorable for the production of the production of the L-asparginase enzyme. So I have choose again the Aspergillus niger. Why? Because you can see here Aspergillus niger is showing the maximum activity as compared to the other sources. So next, I have done the literature survey for my research, and the literature survey reveals that there are so many work which is done on the bacterial L. aspergillus. So I have go for the fungal L. aspergillus because fungal L. aspergillus showing the high activity in my research, as well as there is a few literature survey, few literature is there, few references are there, which is not showing the research on the fungal L. aspergillus. So I have choose the fungal L. aspergillus for the further studies. Yes, after the production, after getting the cultural conditions, optimum cultural condition, I have done the applications of the statistical technique. So in that, Plackett-Berman design is the tool that I have applied for the optimization of the different process variables for enhancement of the L. aspergillus production. Actually, basically, we will do manually, but manually takes a time. Okay, because there are the different carbon sources, there are the different nitrogen sources, there are the different type of the sources that we can use in the media. But this is the statistical technique which will give the result in the small time period. So this is the approach which is called as a one factor at a time approach. So one factor, it means what? One nutrient that we can see in presence of the different type of the nutrient, its activity. For example, I am applying the 10 nutrient at a time, right? 10 nutrient in my class at a time. So in that, which type of the my nutrient is a carbon source. So maltose, lactose, glucose, I am applying in that media. So in that, which type of the sugar that will give the high production, it will give the high production. So that can be identified with the Plackett-Berman design. So here, this is the one factor at time approach that I have used in my further studies. So here I have choose here corn flour, urea, L-aspargine as a substrate. So that is compulsory because as we know that the enzyme will not act when there will be no substrate. So L-aspargine, corn flour, that is the natural substrate that I have used. Glucose, K2HPO4. These are the inorganic organic salts and the carbon sources that I have applied. And after that, I have choose the low level and the high level. So two levels will be applied for that. So high level, it will be like a 1 or 2, and low level it will be the 0.5 or 0.1. So for that I have got the different type of the values, and that values I have applied in the software. So here you can see that this is my design in the PB experiment. It is the Plackett-Burman experiment. So here you can see that. So low value and high value, that will -1 or 1. 1 is the high value and -1 will be the low value. So here you can see that the enzyme activity that I have got in the experiment number 3. So experiment number 3, which is showing the different type of the nutrient that I have used in one time. So this is the result I have got in the Plackett-Burman design. It will be not applied, or it will take some time when we use when we apply it manually. Next, this is the statistical analysis. After that, I have done the t value, p value, SC coefficient. So here you can see that these are the different results that I have got: corn flour, urea, and L-aspargine. So these are the 3 different factors which will show in the positive effect for the L-asparginous production. So this is my conclusion. Next, yes, this is the F value, P value, which is I have got with the Plackett-Burman design. Now, this is a chart that it is showing that whether the combination of the 2 factors, it will give the more efficient result or not. So here you can see that AB is nothing but it's a corn flour and the urea. So AB, that is the corn flour and the urea, this is a combined factor that will give the highest production of the L-asparginase or the activity as compared to the other factors. So you can see here this is a chart that I have got with the statistical analysis. But this chart or this combination I have done experimentally also. So the experimentally the same result I have got as the statistical technique. Now these are the residual plot for the enzyme activity. So here you can see that these are the residual distribution of which is showing the red dots over here. So this will showing that whether the probable or the L-asparginase production it is showing with the different residues highest or lowest. So in that, this is my conclusion. See what different type of the carbon sources I have studied. So here 8 carbon sources, 12 organic nitrogen sources. See, this is very the tremendous task that I have done. 14 mineral salts, 9 inorganic nitrogen sources, and 8 amino acids. So these are different type of the nutrients that I have applied for the production of my enzyme at a one time with the help of Plackett-Burman design. So this is a statistical software which will give the result, which is very valid result, because as we know the statistics which is when we apply, so this will give the validation of the data, and that is very important during our thesis. So this is the carbon sources that I have applied. So I already I have told you that here I have choose different type of these carbon sources, and this is the yield that I have got during my experiment. So dextrin and maltose, it is showing the high production of the enzyme. Here you can see that the values, its coefficient, SS percent T value, etc. Now minus value, what it indicates that actually minus value it indicates that these products are these products are insignificant. Okay, these sources are the insignificant for the production of my enzyme. Okay, so the minus value, the significance of the minus value is that this product, this enzyme will not produce with the help of this type of the carbon sources, because it is showing the insignificant. These are the insignificant, and the lowest value, what it will shows that it will show that it is showing like a large coefficient effect, large positive effect. Okay, but not the higher effect. So higher effect it is shown with the help of the dextrin and the maltose. These are the two sugar which will showing the highest production of enzyme. This is the Pareto chart. Again, the Pareto chart, already I told you that the mixture that we can see with the help of this Pareto chart, and which will identify it with the Plackett-Berman design. Okay. Next is these are the different type of the nitrogen sources. 16 nitrogen sources that I have applied. So here peptone is the more and most highest contribution it will give for the highest production of my enzyme. So here you can see that, and minus, as we know that this will give the insignificant result. So this is the combination of that sources. Here you can see that. Next, these are again the inorganic nitrogen sources. CaCO2, which is showing the high activity. Okay. Next, yes. Okay, so these are my Plackett-Berman or the statistical analysis of my study. Now, next, see, I have done the production with the help of the different enzymes. So in that, I have got the fungal activity more as compared to the bacteria and actinomyces with a different type of the cultural conditions, and with the different type of the statistical the analysis. Now, next, I have done the purification. So purification with the help of the gel filtration chromatography. So here you can see that this is the graph that I have obtained with the help of the gel filtration chromatography. So here, with the help of the Cephadics, I have done the purification of this enzyme. So here you can see that the absorbance at 280 nanometer and the enzyme assay at the 500 nanometer. But still, here there are the two peaks in the first gel filtration chromatogram. But in the second gel filtration chromatograph, there is only one peak. So it states that in the gel filtration chromatograph, second Cephadic G25, this is a more purified enzyme that I have got as compared to the other two, as compared to the first gel filtration chromatogram. And now next, I have done the SDS page. So here you can see that these are the different the 6 lanes. So in one lane, this is the protein marker. Second lane, this is the enzyme, which is the purified with the ammonium sulfate precipitation. In the third lane, crude extract, and in fourth lane, there will be the fractionated. So here you can see that more proper band in the 4 lane. So this is more purified enzyme that I have got with the help of the filtration chromatography. And next, I have done with the native page by using the substrate as an asparagine. So here you can see that the band which appear, there is a presence of enzyme in my purification. Now, purified enzyme that I have applied for the optimization studies. So the pH 8 is which is showing the pH 9, it is showing the highest optimum activity, enzyme activity. Here the 30 enzyme, yes, whether the enzyme is stable at high pH or not. So here you can see here the 80% activity which is retended about the then it is retained, and after that it will decrease gradually. So here you can see that. Next is the optimum temperature. So asparagine activity that we found in the 37 degrees Celsius as the highest optimum temperature for the asparagine activity. Next, then again I have checked the stability of that optimum temperature. So here the 84.23% enzyme activity is showing, it retains the type of the enzyme activity at that temperature. Next, substrate concentration I have studied. So 150 millimolar is the highest substrate concentration. After that, these are the different type of metal ions that I have shown. I have seen out the effect of the different type of the metal ions on the enzyme activity. So here mercury, cupric ions, and the manganese ions, which is showing which is sensitive towards the enzyme in my literature survey as well as in my research also. Next, I have did what is the activity of an enzyme in the human serum. So I have applied without the human serum, the enzyme without the human serum, the enzyme with the serum. So 24 hours I have incubated the human serum with my enzyme. So here you can see that 24-hour, 82% it will retain the activity. After that, it will decrease gradually up till 96 hours. But it will not decrease like the 50%. It means the activity it will get reduced till the 50%. It will not decrease gradually. Next. Yes. Now, yes, effect of different end products. Now, this is actually this graph is showing that whether the enzyme activity is showing the feedback inhibition or not. So, this graph it signifies that there are the two products that is going to form, that is aspartate and ammonia, that will not take again participate again two enzymes, so that enzyme will not showing the feedback inhibition. After that, I have done the immobilization also. Here you can see that these are the some pictures that I have captured during my research. Actually, when this enzyme that will injected through the in vivo, so incompatible assay that we have to study. So it's very important whether the enzyme is showing the incompatibility or not. But in vitro, I have done the immobilization with the help of the different type of the biodegradable polymers. So here you can see that these are the different list of the biodegradable polymer that I have used during the immobilization of the L-asparginase. So sodium alginate is showing the more or enzyme activity as compared to the other type of the carrier. Now I have done the KMV max also. Now, next part is what the validation. Structure validation. So, bioinformatics that I have applied for my research, it is showing that actually what is the active site of my enzyme on which the substrate it will bind. So actually, this is not our part because we are the biotechnologists, and this is the statistical technique that bioinformatics technique that I have applied with the help of different type of resources. So thanks to Dado sir that he have uh, he had guided me at that moment. So in that, what I have did, I have like 10 minutes I will tell you. So in that, what I have did, um, uh, actually the structural prediction cannot be done in uh, lab or in a test tube. We don't know what type of the structure of the enzyme it is having, but with the help of the statistical technique in which bioinformatics technique, that is Geno 3D software, which is the prediction software that I have used. I have predicted the enzyme structure which I have isolated with the fungus, that is Aspergillus niger. I have compared that structure with the E. coli, which is available in that software. Okay, so Geno 3D is the structure prediction software, and in that software there are the different type of the enzyme structure is available. You can see online. So here, um, yes. So here you can see that this is the structure of my enzyme. That is the L-asparginase, which is I have isolated with the Aspergillus niger. And these are this is the enzyme which is already isolated, which is inbuilt in the bioinformatics software, that is E. coli. So here you can see that the comparison, there are the so many compact of the binding pattern is there as compared to the E. coli. The prediction is very difficult with the fungal because fungal cell is a eukaryotic cell and it will not predict it very easily. Next, yes. So docking. What is the docking? Actually, docking is nothing but the interaction between the enzyme and the substrate. So enzyme substrate interaction we cannot be predict with the test tube, with the in the lab, but we can predict with the help of the software. So here the software that I have choose for ProCheck software. So with that ProCheck software, I have done the docking. So these are the Ramchandran plot. Here you can see that the favorable regions, the residue where they are present in that favorable region. Next, ligand. So the ligand is nothing but my L-aspargine. So L-aspargine that is also predicted. The structure of the L-aspargine is predicted with the PubChem compound database software. So here you can see that this is the structure, and these are the structural information I have got with that software. And these are the results which is showing that docking studies, whether there is interaction binding of that enzyme with the aspargine is whether the binding is occur or not. Okay, so here asparginase enzyme which is from the fungus, and this is the L-aspargine. So here you can see that which type of the interaction that is happening here in case of the fungal L-asparginase, and this is in case of the bacterial L-asparginase. Now, so type of the bond that I have studied. So in my study, in my statistical and as well as the bioinformatics study, what I have seen that the binding pattern, H binding, H bond, ionic bond, the van der Waals force of attraction, pi interaction is more in as compared to the bacterial isolate. So here is you can see that. Now, the low binding energy values that will indicates that the ligand is most favorable region of the protein. So I have got very low binding value here. You can see minus 3.35. So it means that my enzyme is very proper. Actually, L-aspargine enzyme is very proper that I have isolated with the fungal culture, which is more favorable to its substrate or to its ligand. Next is okay. Now now next part is nothing but in vitro study. So in vitro studies application part actually. So in vitro studies I went to the Tata Institute, which is situated in the Kharghar, Actrec. So in that there the Cancer Institute is there. So in that I have studied the extracted enzyme which is purified with the gel filtration chromatography, fungal L-asparginase, that I have applied with the different two cell lines, that is leukemic cell lines. So I have performed the LAL test for that. So this LAL test signifies that whether the enzyme which is isolated, which is showing the presence of the endotoxin or not. So the LAL test that I have performed. Next, cytotoxicity of L-asparginase preparation. So in that I have performed the two, that is MTT and SRB test. So here I have got this type of the result. So the drug dose required to cause the 50% reduction in the cell viability. So viability that I have checked. So here you can see that the result, and the two cell line that are used, MALT4 and MCF7. So human breast cancer cell line I have choose because this is a more favorable cell line which is showing the highest positive effect with this isolated L-asparginase. So these are the results that I have got with the Tata Institute. Now, so from that, I have concluded that the whatever I have done the research in 2009, actually this is long back, that is showing the highest enzyme activity in case I have choose the fungal as a bacterial fungal as a source for the L-asparginase production. It is showing the highest enzyme activity as compared to the other different sources, and that is showing the strong anti-proliferative activity as well. So that I have concluded that it is more superior than the currently used dose or the currently used the vaccine which is produced by the bacterial L-asparginase, because bacterial L-asparginase is showing the higher hypersensitivity reaction or the side effects. But side effects cannot be shown with the fungal aspergillosis that I have isolated. So the future task that during my research that will be the scale up and in vivo studies, because in vivo that I have not applied because this is for the like animal that is not actually the ban was there. I have not got the animal for in vivo studies. But still, I have done the in vitro studies, and in vitro it will give the more proper result as we have seen here. And after that, molecular level studies that I have I would like to do in the future aspects, and the production of the cloned asparagines by the recombinant DNA technology. So these are the future aspects of my research, and now I'm doing in the my research laboratory in the MDM. Because L-asparginase it is not showing not only as a therapeutic enzyme, but it is showing the application in the reduction of the acrylamide in the food preparation, in the fried food preparation. So nowadays I'm doing the research on that. So this is done from my side. Okay, sorry for interruption. So this is from my side. Thank you so much for giving me the opportunity to talk to you. Thank you. Yes, Krishnanjali ma'am.
Hello, ma'am.
Yes, ma'am.
Oh, okay, you're done, ma'am.
Yeah.
Oh, thank you, ma'am, for your presentation. Now we'll start our question answer sessions. So, audience who have doubt, they may ask question by texting, or otherwise you may ask directly one by one.
Good
Yes.
evening, ma'am.
Yes. Good
There are
evening.
few questions in chat box. Should we
Yeah.
take that first, ma'am? The
Yes.
question
Yes.
is from Dr. Vinay Aroskar. Did you use soya flour in media? And as there are reports that soya enhance active asparaginase enzyme.
Yes. Yes. Actually, soya flour that I have not used, but yes, that also use that act as a natural carbon source to enrich the L-asparagine enzyme activity. But I have done the project, actually small project on that. The In that Priyanka Patil that actually she joined my just now she have joined, actually she have joined my presentation. So Priyanka Patil she have done the research, the project on that, and she have done the soya flour in the media. Not only the soya flour with uh, the Mosambhi bagasse also there, Mosambhi bagasse, and other natural resources which is called as the industrial waste. So only industrial waste we use here for the hash production of the enzyme activity. But soya flour that also we have done, but it is not in my research, but we have done in the a small scale of our project. Yes.
Uh,
And next will be the whether pH 9 also prevent the growth of aspergillus niger. Yes, actually, fungal that will grow in the acidic condition, but a pH 9, it will give the more proper result in my research study. So this is the controversy that I have found actually, which will give the change in coloration, because I have used the phenol red as an indicator, and here there will be the change in coloration, and that I have seen in the enzyme activity. Next is the may I know title gram per liter as economical is above 1 gram per liter industrial scale. Gram per liter. Industrial scale, actually, okay. So you want to scale up. So 1 gram per liter. So if you want to scale up, so if your titer is having the 1 gram per liter, so is it, yes, it actually the question is I'm not understanding. Dr. Vinay, uh, Dr. Vinay, am I audible to you? Dr. Vinay, are you there?
Can
Hello,
you hear me?
Dr. Vinay sir.
Can you hear me?
Hello.
Yes, can you
Yeah,
hear
yeah,
me?
yeah,
Yes,
yeah,
yes, sir.
yeah, sir. Continue, sir.
Yeah, what,
So
may I know that
what's your
tighter,
question?
what tighter you got? What tighter you got?
What title?
Yeah, per gram per liter we generally express in our
Uh-huh.
fermented drug. So what
Uh-huh.
tighter you got? What is the activity you got?
So you are asking in the IU per इंटरनेशनल यूनिट में आप पूछ रहे हो या एमजी पर एमएल?
Both. If you have both, okay.
हाँ,
Otherwise, IU per ml.
yes, actually point one, sorry, 0.5 mg per ml, 0.5
Okay,
mg per
fine.
ml. Yes,
Okay,
so you can convert per liter, right?
okay,
Yeah.
okay.
Yes.
Yeah.
Okay, there is
And
one
which
more
kit
question.
did you use? Which kit did you use for endotoxin?
Sir,
Which
pardon, sir.
kit did you use for endotoxin determination?
Lal test I have performed actually. Lal
No, which
test.
kit did you use? Which kit? Whether it
Heat,
is Charles River or
actually that was done in the Tata Institute. There, that actually you know that all the institute which is accredited, they will not show. They will directly give the result in our hand.
okay, okay, okay.
Ha ha.
I got it.
So
Okay.
yeah.
Because I work on this
Right,
one,
right.
Okay.
donomycin. I
Yes,
work
ma'am.
on donomycin
Okay.
fermentation.
Okay. Okay. Nice.
Okay, we'll take the next question, ma'am.
Yes.
It's from Abha, ma'am. She wants to know about the enzyme assay method used and emphasizing on buffer and substrate concentration as well as control.
Okay. So in the enzyme assay that with the help of Nestler's reagent, I have applied the Nestler's reagent for the ammonia estimation because ammonia acts as a byproduct, and in that I have done the colorimetric or the absorbance. I have seen the absorbance and the 500 nanometer the reagent. But during the assay, what I got the precipitation in my suspension. So to avoid the precipitation, I filtrate the suspension, and I have done. I have did uh, the uh, the assay as well as the enzyme activity. So there is a formula enzyme assay and for the enzyme activity as well. Yes. I think all questions that I have resolved.
Yeah, I think so. Completed. Anyone having doubt, they may ask questions.
Good evening, ma'am.
Yes, very good evening.
It was a very nice presentation and good piece of work that you have done.
Thank you so much.
Congratulations for it. Ma'am, I
Thank
just
you.
had a doubt regarding the anti-proliferative activity which you have done for the enzyme. Okay.
Acha.
Huh.
Actually, anti-proliferative activity that I have done with the help of MTT and SRP. So MTT assay that I have applied, so actually kit that I have used. So basically, that is also done in the Tata Institute. So in that, the MTT assay, the MTT buffer they have prepared, it is having a different type of constitution, and my enzyme that different constitution with that buffer, and the cell line. So they incubate that, they have incubated this type of thing, and they have seen the activity, they have analyzed the activity with the help of the spectrophotometer. So that they got the result that they got the result that with the help of fungal L-asparagines, which is I have isolated the MTT assay or the anti-proliferative cell line assay, because uh, anti-proliferation of the breast cell line that I have got in that assay. So which is showing the highest activity as compared to the other cell line, because at that time the cell line are very expensive. So actually I have done with the breast cell line and the HeLa cell line. So in that, in two cell line, I have got the MCF HeLa MCF cell line, which is the breast cancer cell line, more anti-proliferative activities as compared to the other. So this is with the help of MTT and the SRBS.
Okay. Thank you, ma'am. Ma'am, one more question. The
Yeah.
purified enzyme which you have got after various gel chromatographic techniques, how did you maintain it in a stable form in the lab? Because usually the enzyme loses its stability upon long-term preservation. So
Yes.
what was the thing that you had done for maintaining the stability of the enzyme?
Actually, I have yes, this is a very good question. As we can stable the culture with the help of different preservation technique. So the enzyme will not be preserved for the long time. Correct. Okay. So for that purpose,
या सरळ आहे ते ऍक्टिव्हा पासून सरळ आहे.
okay. So for that purpose, what I have did, I have used the ice bags, and within 8 days, actually, I have seen. I will see. I had checked the activity for every day. Just I have checked activity today, again day after tomorrow, day after, like that. So I have checked the activity of the enzyme every day. Okay. So there will be like a minor 0.0 something negligible value that will decrease day by day. Okay. But when I incubate that enzyme, or I preserve that enzyme in the -20, so that will not reduce the any type of the activity. And at that time only, I have given the sample to the Tata Institute with blue dart, with the blue dart. So within 8 days, this can be preserved in the ice bag in the -20. Yes. So I have not used any type of the preservation and all.
Okay. Thank you. Thank you, ma'am.
Hello.
Yes.
Okay, any questions from audience side? Hello. I think it's done, ma'am.
Yes. Okay.
So it's time to now thank you everyone. First of all, I thank you, ma'am, for giving your time and for your presentation. And also thanks to AM Deshmukh sir and all other participants. Thank you everyone for joining us. And if any doubt, ma'am, you may share your email ID. So if anyone having doubt, they may
Yes.
contact you through email.
Yes. Yes, of course. So thank you so much by giving me the opportunity to present in front of you. Thank you, Nitin Adapure sir, and thank you, Dr. A.M. Deshmukh sir also, who is the organizer and who is the motivator of all of us. Thank you so much. Thank you, ma'am.
Okay, thank you, ma'am. Thank you. Ma'am, you may leave.